You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health podcasts, and I have Tamer Mohamed. He's the CEO of Aspect Biosystems, and their website is aspectbiosystems.com. So, Tamer, thank you for being here.
Thank you for having me.
Yeah, so tell me about Aspect. What's the premise of the company?
So Aspect is a biotechnology company. We're based in Vancouver, Canada, and we're working towards enabling the creation of human tissues on demand using our 3D printing technology.
So human tissue, just to give people an idea, how many different, first of all, what constitutes a tissue, and then how many different types of tissue are there in the human body, for instance?
Yeah, so that's a good question. What we've developed at Aspect is a platform technology. So we spun out of the University of British Columbia here in Vancouver about 5 years ago. That technology that we brought into the company was a broadly applicable 3D printing technology that is capable of producing a variety of different tissues. And so our focus at Aspect has been to use that platform to create tissues for drug development, so building tools for therapeutic discovery, as well as transplantable tissues. Some of the tissues that we're focusing on internally in the company include knee meniscus tissue for transplantation. So this is a fibrocartilage tissue in the knee that is very commonly damaged. We're also developing a portfolio of muscle tissue, including intestinal muscle tissue, cardiac tissue, skeletal tissue, and smooth muscle tissue. We have a liver program that is focused on developing a liver drug testing platform for pharmaceutical discovery, as well as a pancreatic tissue program for the treatment of type 1 diabetes. But we also work closely with academic partners around the world. We give them access to our platform to develop other applications. We see this really as our innovation pipeline. And so we have researchers around the world focusing on kidney tissue, skin tissue, vascular tissue. And so this is really our strategy around trying to unlock the broad applicability of our platform without defocusing internally. We like to work with our strategic partners around the world.
Yes, I've spoken to a few people about 3D printing, organs or tissue. I guess some of the major issues is that you have to make sure the tissue is vascularized. I guess what's the maximum distance that any given cell can be away from like a blood supply in order for it to live? It's a metric, I think it's like a millimeter or less, or I don't know. It seems to be that a big roadblock to doing this.
Yeah, it's a good question. I think when you look at sort of what's been reported on the literature, you'll often kind of see that there's a couple hundred micron sort of limitation, and really what that is kind of about double the thickness of kind of a human hair, just to put that into perspective. But we've been focusing on really innovative strategies to vascularize our tissue. And so we have the ability at Aspect to create what we call hollow fiber technology. And so really what this allows us to do is to pattern in hollow features within our structure so that we can run materials through these tissues and ultimately not just keep them alive, but simulate a more physiological type environment. And so an example of us using that technology is through a partnership that we announced in November of last year with JSR in Japan to create vascularized liver tissue. And so we'll be using this technology to perfuse essentially living functional liver tissue. So that's one area that we're actively sort of employing our vascular technology, but we also have other strategies in the company, and we collaborate with folks that are interested in this area and have expertise in this area. One example of that is a partnership we have with UCLA in California, where they have access to our platform and are essentially developing vascular technology. But there's also a lot of other tissues in the body that aren't well vascularized, which makes them not tissues that are good when you have an injury, but also makes it easy to, I guess, tackle from an engineering perspective. So an example of that is the meniscus tissue. So the meniscus tissue is not a very well vascularized tissue. It's a tissue that is one of the most commonly damaged parts of the knee, and then once the damage occurs, it doesn't get better over time. It just gets worse and worse, largely in part because it isn't well vascularized. And so current strategies include meniscectomies, where a surgeon will go in and try to remove the damaged part, or in some extreme cases, they'll remove the entire part of the entire meniscus. That'll alleviate acute pain, but often introduce arthritis in the knee. And so that's an example of an application where we believe the science is there. We believe our technology brings unique value, and it's not a vascularized tissue. And so that's a program that was conceived with Johnson and Johnson, and we're very excited to move that program forward and ultimately try to make an impact on clinical care with this.
So what's the hierarchy? What's the easiest, lowest hanging fruit tissues, which I know is a weird metaphor, but and then what are the harder ones and what's like the hardest ones that probably will take the longest to get anywhere to make?
Yeah, at Aspect, we kind of really don't think of it in terms of a future where we will be printing large functional organs, and we're at a path towards trying to do that. Our goal really is to create advanced living tissue therapeutics, and what I mean by that is we might not have to recreate an entire organ just like we understand these organs in terms of how they look and how they function exactly. What our goal is to do is to use living biology, use our 3D printing technology to almost create these smart living devices that we could implant into the body to essentially take over function that has been lost. And so from that perspective, there are a lot of possibilities. So the meniscus is one example. Another example of a program that we're working on is a pancreatic tissue patch. And so this is a patch that will be vascularized, would include insulin-producing beta cells, and would be targeted for patients with type 1 diabetes. So that patch will look nothing like a pancreas, but it will provide a very necessary function. All of these opportunities have their own challenges, but we believe kind of the goal of creating these living tissue therapeutics is a much more realistic vision than the vision of trying to create a lung that looks exactly like a lung, or a heart that looks exactly like the heart and is the same size of the heart. And so that perspective in mind, we actually see a lot of opportunities right now that we're targeting, and we always start with the question of where is the area of unmet medical need, and then work our way backwards to where our technology could provide value. That's kind of our perspective on that.
All right. So let's get into maybe some mechanics. So when you're going to 3D print tissue, are you printing a scaffolding and then you're placing cells as you print, or you just do a scaffolding first? Like mechanistically, how does it work to produce various tissues?
Yeah. So the traditional approach to tissue engineering involves doing exactly what you described, which is build a scaffold first and then seed cells on top. And there are certainly opportunities with that strategy, but there's also limited control. You can imagine if you're trying to create a multicellular type construct with heterogeneous properties, it would be very difficult to do that if you employ kind of a strategy like this. And so what we do at Aspect is we've developed a microfluidic technology that allows us to take in multiple different input materials. So these are cells, scaffolds, growth factors, extracellular matrix content. We take all of these compounds and we could combine them to form this cell-loaded fiber that we pattern in 3D. to create our tissues. And so the cells are actually embedded in the scaffold. They're embedded in this biomaterial fiber that we dispense and pattern in 3D. And so we're able, through this microfluidic approach, to really create this multicellular, this multi-material heterogeneity that is really important in tissue engineering.
Yeah, and how do you achieve vascularization? If you seed, a scaffolding with cells, and then the cells start growing into it, how do you control where the cells will go and where they won't go, and how do you make sure they leave room, and the vascularization, how do you make sure it occurs along the tubules or the paths you want it to occur, and not just not occur or randomly occur in the wrong spot?
That's a good question, and so one sort of embodiment of our technology involves not creating solid fibers, but creating hollow fibers, and we can actually cellularize the shell of that hollow fiber. And so what comes out of our printer is actually a blood vessel-like construct, and we can pattern that by design. So that's one strategy. The other strategy is relying on biology, like you mentioned, to do the magic in terms of seeding cells in the right environment, or putting the structure in the right environment and letting that vascularization manifest. And so I think ultimately it'll be a combination of both, putting in the right blood vessel-like features, providing the right environment, the right growth factors for the overall kind of vascularization strategy that is necessary.
How often have you, put cells onto a matrix or a scaffolding, and then, let them grow on their own, and they've done all kinds of interesting patterns, or seem to have a behavior that was, I don't know, surprising or amazing to you? Like, do cells seem to, I mean, they must operate by a code that we don't understand, is what I'm saying, in order to build an organ with a particular architecture. So do you see that when they're placed onto a 3D scaffolding, that they behave in predictable ways? And I mean, what does that look like? Can you say more about that?
Yeah, that's a great question. I think really what we like to look at 3D bioprinting really as 4D bioprinting, with the fourth dimension being time. After you print your construct, all you have done is built a tissue precursor. In the sense, you put in all of the right ingredients in what you think is the right location, but you really need time and the right conditions to sort of take your tissue to where you want it to go, or where you think you need to go. And so that is often the case with all of our tissues. Is we'll set up a multicellular tissue precursor, and we'll let it develop. And sometimes we have to go back to the drawing board and sort of change our tissue precursor to get the intended result. But it's really beautiful to see how the cells really do their magic. A lot of times it is magic because we may not understand it all. I think our understanding is getting better and better. There may be a day where we could understand that transfer function from printing all the way up to tissue has developed. One example tissue that we've developed that I think is a good example of this is our Airway smooth muscle tissues. And so in this project, our goal was to work closely with a pharmaceutical company to build a model of airway smooth muscle contraction for their asthma program. So in this project, we printed these tubular structures about 5 millimeters in diameter, a couple millimeters high, and it was printed using primary airway smooth muscle cells. So these are cells obtained from real donor organs. And so these are the cells that wrap around their airways, and during an asthma attack, histamine is released, and it causes these muscle tissues to contract and reducing airflow, and so asthma patients have trouble breathing. So we printed these tubular constructs using those very same cells. Then we printed them. It takes about 30 seconds to print, and we let them develop over time, and we could start to see the muscles actually forming really beautiful alignment, circumferential alignment, and when we add compounds like histamine, we can actually see the tissue contract, and we can measure that contraction. Then we took anti-asthma therapeutics and dosed these tissues, and we began to see them relax. And so that's an example of immediately after printing, we won't get that response. But putting these tissues under the right conditions and leaving them to develop over time, we start to get functional tissues, and these tissues are providing real valuable insights in the development of novel therapeutics.
So what are some of the surprises you've encountered in your work, things that you saw happen that you're like, wow, that's crazy?
That's a good question. I think, we're consistently, I guess, pleasantly surprised. I think one of sort of the, the powers of our approach is we essentially have many degrees of freedom to integrate many different types of cells and many different types of materials. And so what that really means is we start to see some interesting interaction between these different materials. An example of that is in the meniscus tissue. So the meniscus tissue is not a homogeneous tissue. It's made up of many different types of materials. And so we've seen that if we try to print that construct with a single material, we don't get the necessary mechanical properties that are necessary for the function of that tissue. But when we start to employ different materials in different regions, we start to get the necessary tensile and compressive strength that is so important for a tissue like the meniscus. So I would say I've always been pleasantly kind of surprised to see our technology demonstrating the value of multi-material capabilities.
So is every tissue in the body more complicated than first meets the eye? Is that the message here?
It is. Tissues are complex, and we need sophisticated technologies to attempt to recreate these tissues. If you look at something like the lung, it contains over 40 different cell types, all highly differentiated. It contains different cell densities, different materials like ECM and growth factors. So we really need a technology that could handle all of these different types of materials. What you get with 3D printing is structural design flexibility, but you don't necessarily get compositional design flexibility. That's what we get with the microfluidics. What microfluidics is really good at is manipulation of materials and fluids. And so by marrying microfluidics with 3D printing, we get a technology that is flexible, not just from a structural perspective in terms of creating very complex architectures, but also a compositionally flexible platform. And that's really important in tissue engineering, more so than just standard 3D printing, where you could create a variety of different products with a single material.
Are you able to, using time lapse and using other assays, to study how cells integrate themselves into a 3D matrix that you'd create, or is it hard to get the resolution on what's going on, like how the cell is preferentially dividing and differentiating and going in one direction versus another and making patterns? And have you been able to observe cells as close as you want, and have you seen any tricks or tips that's helping you with the 3D printing side of it?
Yeah, it's a standard part of our validation of our tissues and monitoring kind of the development of these tissues. It's amazing how far microscopy and imaging has come to the point where we can really observe tissues at the lowest of levels. And so that's a standard part of our validation. We also look at other ways of monitoring development and function. We look at genomic analysis, phenotypic analysis. We'll look at functional endpoints. So, for example, like the meniscus, our goal was to look at tensile and compressive strength. So we actually built simulators that actually allow us to essentially simulate different forces and measure that. With the airway muscle tissues, we can look at these tissues contracting using video microscopy. I think because of that dimensional part of what we're doing here, that temporal dimension, it becomes important to, like you said, monitor changes over time, because it is in a static environment.
So what's possible right now in terms of tissue engineering, and what's ahead in the near future, the far future? So the near being what's possible now, then maybe in a year from now, and then maybe 5 years from now. What do you see as happening?
Yeah, so I mean, technology like bioprinting is only as good as the science that we understand, science of stem cell engineering and biomaterials. And so the great news is we understand a lot about those different areas. And so as a result, there are a variety of different applications that we're pursuing at Aspect and others are pursuing that are really exciting. Things like the meniscus, things like the pancreatic tissue patch, using tissues for drug development. But the even more exciting thing is a technology like ours is just going to get better and better over time. So I think in the future you'll start to see very complex multicellular sort of tissue therapeutics that are providing multifunctional sort of purposes inside of the body. Maybe the first set of tissues will be single function, something like the pancreatic patch that monitors glucose in the blood and then releases insulin as a result. But maybe in the future we could start to see multicellular or multifunctional type tissues that are doing much more than that. And so I think the future for a technology like this is very, very exciting. I think what the industry needs more than anything is success stories. So I think it's very important to use the science that we have right now to develop real tissue products and use that as a success story that could give the whole industry confidence to tackle on more and more, rather than choosing maybe the most complicated opportunity where the science may be not fully understood and risk a catastrophic failure, for example, that could set us back. So for those types of applications, we work with our academic partners through what we call our academic discovery ecosystem. We let the science mature and develop, and once we feel it is ready to enter into more advanced commercial stages of development, we work with our academic partners to do that. And so, for example, we have an opportunity in the kidney space. It was a collaboration launched at the University of Maastricht. This is an area that we think there still needs to be some incubation and academic sort of maturation of that application before we decide to take that on with our academic partners. And so that's why we pursued that in the research space. Yeah.
Right. So again, I don't mean to put this very crudely, but is one way to look at what you do like a tissue band-aid? You know, like the pancreas would be like a beta cell band-aid you slap on the pancreas and now you can produce insulin. And, the meniscus, would that be I guess like a mini transplant or a slathering on of or an implant of a meniscus tissue that would grow and make a new knee meniscus? Is that a way to put it?
Yeah, that's definitely one way to put it. But no, I definitely like how you're thinking. It's really what we're doing is we're printing advanced living therapeutics or devices, or you could call them bandages. But I think definitely our goal is not just to create large functional organs, just like we understand them and know them from inside of our body. It's exactly kind of what you're talking about.
Is it? Do you think you can improve upon what's in the body, or is it so complicated and so evolved that we should just be happy to approximate it or copy what's going on in the body in a given tissue?
Yeah, that's an interesting topic altogether. I think if we can do what evolution and what biology is capable of doing, I think there's just so much more value there. And so that's our target is how do we use biology? We see biology as our friend, so we don't want to start forming enemies with biology. So biology is our friend. We're trying to use biology right now to help people. So that's the focus.
Okay. And then I know this is just guesses, but is any of this technology in clinical trials, and will people be able to have these therapies in the near future? Like, will I be able to get a meniscus? Can I get a meniscus grown and put into my knee? Like, I tore my AC 20-some years ago. So an example of what you're talking about. So do you think in the next few years, or it's going to be longer, that I can get for instance, a meniscus transplant of something that's grown in the lab?
Yeah, and I mean, your case really touches home, as with many others. Like, these are real world things that we're working on, and the impact could be pretty big on people that we all know in our inner circle. Even everybody, especially here in Vancouver, people are often falling down the mountains on the ski hills, tearing their meniscus. And so it really touches home for us. We are several years away. This is still in development, and it's very hard with things like biology to give very deterministic timelines. But I definitely think within the next decade, we will start to see commercial and clinical realizations of 3D bioprinting. So in the biotech world, that's like tomorrow, and so that's what's exciting.
All right, well, very good. So, what's the best way for folks to reach out and contact you and get more information? Do you have a video on 3D printing looks like, or how cells grow into a matrix, or any other resources for listeners that want to learn more?
Yeah, the best way to learn more is visit our website, aspectbiosystems.com, and we have material there and access to our social media channels and videos, and definitely reach out. And those would be the best ways to contact us.
Okay. And then I had to make one joke, but in the future, if you're crying, you can get a tissue for more than just your tears. You can get a tissue to fix your body. It's my bad joke.
That's a good one. I may have to use that.
All right. Well, very good, Tamer. Thank you for coming on the podcast, and I really appreciate it.
My pleasure. All right. Take care.
You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you.